Jambriupsu, everyone. My name is Talia Moore, and today I'm going to be telling you about which statistical test you should use. First, a little bit about myself. I'm an assistant professor in the departments of robotics and mechanical engineering at the University of Michigan. I've been working with RUPP since 2019, so some of you may have met me. My expertise is in biomechanics, which is how animals move, evolution, experimental design, and bio-inspired robotics. I've worked with rodents, snakes, lizards, and insects. Okay, before we get started, I need to tell you some statistics vocabulary. Discrete data are data that are described by categories or levels. So these are data that you describe with words. Examples might be color, blue, green, red, yellow, sex, male or female, what country you come from, did something survive or not, was the test completed or not, these sorts of things. Continuous data can be measured with numbers. So continuous data are the opposite of discrete data. Continuous data can be things like age. How old are you? The answer is a number. Size, again, a number. Length, temperature, time, mass, all of these are numbers. These types of data are very important for finding the right statistical test. We have a little bit more vocabulary before we dive in. So here we go. Factors. Factors are inputs into your experiment. So for example, maybe these are different rivers where you're sampling data. Levels are the variations in the factors. So you want to measure the effects of these variations. So maybe you have the Tonle Sap and the Mekong. Those are two types of rivers that you have in your experiment. So the factors are rivers and the levels are Tonle Sap and Mekong. The responses, the output of the experiment. What are you measuring? So maybe you're measuring how many fish can I catch in each of these rivers. So the thing you would measure is the number of fish. In this example, the number of fish, that's a number, right? So this is an example of continuous data. The Tonle Sap and the Mekong, those rivers, those are discrete factors, discrete levels of your factor. The null hypothesis. Every type of statistical test has a specific null hypothesis. And what this means is it's this assumption that you're trying to disprove with your data. So the null hypothesis usually has something to do with no effect. So no difference due to the variation in the factors. For example, in our situation here, my null hypothesis would be the Tonle Sap and the Mekong River have the same number of fish. An alternative hypothesis is the opposite. So if you disprove your null hypothesis, then the alternative hypothesis must be true. And the alternative hypothesis usually indicates that there is a significant difference due to variation in the factors. So in our example, the alternative hypothesis would be the Tonle Sap and the Mekong River have a different number of fish. Okay, these are the tests that we're going to cover today. And the way that I've organized them in this table is by the type of factors and the type of responses. So remember the factors over here on the left, this is what you're trying to find a response to. So these might be your different rivers. These might be different levels of drugs that you're inputting into the system. And your responses, remember, these are what you're measuring. So depending on whether your factors are discrete or continuous, and whether your responses are discrete and continuous, this will determine which is the right test for you. Okay, now I'm going to go through each of the tests one by one. The first test is the Fisher's exact test, or the chi-squared test. I'm mainly going to cover the Fisher's exact test today. The null hypothesis for the Fisher's exact test is that two categories cannot predict two other categories. Here is an example. Does the medicine cure the disease? This is our question for research. How do we collect data for this? We would arrange our data in a table. So here we have the number of people who took the placebo. Remember, that's a pill that has no medicine in it. That's kind of our control for our experiment. So the number of people who were cured when they took the placebo is 20. The number of people who were cured but who took the medicine is 487. Now, the people who were not cured, who took the placebo, is 480. And the people who were not cured, who did take the medicine, is 33. Okay, so what do we do with these data? One thing to consider, the reason why I am covering the Fisher's exact test more in detail than the chi-squared test is because the Fisher's exact test is better for small sample sizes. And usually for the type of research we're doing for a master's thesis or for cool projects, we'll usually have smaller sample sizes. So the Fisher's exact test is probably more appropriate. It's probably the one you want. But I want you to be aware that the chi-squared test exists. It's better for large sample sizes. And the chi-squared test also looks like this. This is the symbol that you use for the chi-squared test. Okay, now what do we do with these data? I'm going to be showing all of my examples in R. So R is a statistical programming language. The way that you would make a plot of your data for Fisher's exact test is using this mosaic plot. So this shows you how many people were cured who took the placebo and who took the medicine, and how many people were not cured who took the placebo versus the medicine. So this already visually shows you the kinds of trends that you have in your data. So the mosaic plot, if you're doing Fisher's exact test, is a good one to put in your report. These are the lines of code that I wrote to create this mosaic plot and to run the Fisher's exact test. So the Fisher.test and then study has the data from the study, right? So study is a data frame. I added the number that were cured, the numbers that were not cured, and then I showed that the row names for cured were cured and not cured. And then the column names are placebo and medicine. So here I have our mosaic plot. This is mosaic plot. That's the function that you use to create this plot. And then I have the Fisher test. When I look at what is given in this Fisher test, it tells me, okay, this is the Fisher's exact test for count data. This is the data set that I gave it. The p-value is 2.2 times 10 to the negative 16th. So the alternative hypothesis here is that the true odds ratio is not equal to 1. That means that they should be the same. And what this means is that this p-value is much less than 0.05. So that means it's very, very significant. So you know that in this experiment, the medicine does cure the disease. Okay. Next, I'll talk about the student's t-test. In the student's t-test, you have data from 2 different sources, and your null hypothesis is saying that the sources, the data from those sources are the same. And here we're talking about discrete factors, and we're talking about continuous data. So for example, do 2 rivers have the same level of pollution? The factors are the 2 rivers, and those you have to describe with words. And then the level of pollution is something you measure with a number. So this might be what our data look like. Let's say that these numbers represent parts per million of a pollutant. You look at river 1 and you look at river 2, right? These are basically just 2 different names for the rivers. What do we do with these data? Well, first, what we need to do is create a histogram. So with the histogram, what you see is the frequency on the y-axis, the frequency of each level of pollution. Okay, so this tells you this is how the data are distributed across those 2 different rivers. And what I've done here is create a line, a vertical line, to show you the mean of each population. So here's the R code that I used to create this plot. I input the data for river 1. I input the data for river 2. First, I made a histogram of river 1. And then I added the histogram for river 2, and I added it in blue. Here I have the line that shows the mean value for the river 1 data. And then I add the mean value for river 2 as a vertical line in the color blue. In the next line, I'm running the t-test. So I say river 2 and river 1, are they the same? And when I ask the computer, what's the result? It tells me this is a Welch 2-sample t-test. The data are river 2 and river 1. It tells me the p-value is 0.0057 something. So what this means is it has rejected the null hypothesis that the true difference in the means is equal to 0. So it supports the alternative hypothesis that the true difference in the means is not equal to 0. So we have our p-value here. That is 5.8 times 10 to the negative 3. That's much less than 0.05. So it is significantly different. Okay, next, let's talk about the ANOVA. This is very similar to the student's t-test, but it's looking at 3 or more sources. So the null hypothesis is that data from 3 or more sources are the same. For example, you might ask, do all rivers have the same level of pollution? Here we have example data from rivers 1, 2, and 3. Remember, these are discrete because they are discrete rivers. These are the names of different rivers, and the data are continuous. They're the amount of pollution that you are measuring. Okay, and remember, if it was just 2 rivers, you could use the student t-test, but because we have 3, we have to use ANOVA. So what do we do with an ANOVA? With our data, we can plot them with a box plot. This is what the box plot looks like. We have River 1, River 2, and River 3. It shows us how the data are spread. So this line shows us the median value for River 1. This box shows us the edges of the quartile, so where most of the data are between. These show us what are the extreme values, and then if there are any outliers, they're represented by this circle. Usually, if we see that these boxes are not overlapping in space with another box, it usually means that they are significantly different from each other. So just from this plot, I can guess that River 2 is significantly different from Rivers 1 and River 3. How did I make this plot? This is the R code that I used to make this plot. So I added the data for River 3. Remember, I already had the data for Rivers 1 and 2 from the previous example. I told it what the groups were, and then I turned it into a data frame. So I compiled all of the data together for all 3 of the rivers into a data frame with this group value. And then AOV stands for analysis of variance, or ANOVA. So I say, I'm asking it, okay, between these 3 rivers, the data that are in the 3 rivers, is there an effect of the group? So when I ask for a summary of that, it will tell me, okay, the p-value is 0.0009. And that tells me this is very significant. And then I can ask for a box plot of the same data, and it will give me the box plot. Okay, next let's talk about logistic regression. Logistic regression is when you're trying to use continuous data or number data to predict a category or a discrete response, right? So you're using numbers to predict a category. For example, you might ask, are larger snakes more likely to bite people? So the size of the snake you measure with a number, and the bite is a yes or no. This is not a number. So here's our data. We have snake size here, and then we have bite. Did it bite? Yes or no? For logistic regressions in R, we want to make a logistic regression curve. This is the plot that we would use. So this tells us the first thing we need to do is change the bite yes or no into 0 to 1. And this gives us the probability that a snake will bite or not. And then we'll say, okay, we see that these snakes with these masses did not bite. And these snakes with these masses did bite. And then this green line gives us a prediction of how strongly we can predict whether or not this factor has an effect on the response. So does snake size actually predict the bite? What you really want is for there to be a sharp curve, okay? This is a very gradual, smooth curve. If you had a curve that looked very sharp, like an S shape, that is going to be more significant. This diagonal line is not very significant. How did I make this plot? This is the R code to make this plot. So I added the snake size data, I added the bite data, and I wanted to make sure that the bite is a factor. So then I put those together into a data frame. And then for plotting, I needed to do something slightly different. I needed to sort the snake size data by size, so going from smallest to largest. And then I needed to change the bite data to a 1 or a 0. Even though it's a number, it still is representing discrete data. So then I put those together into a data frame for plotting. I create a model. This is a generalized linear model, but I use the binomial family to make it a logistic regression. The binomial family means it's just ones or zeros. There's nothing else, yes or no. Okay, so then I fit the model here, and then I use the model to predict the data. So it's built a model, and it's going to tell me how is this model predicting the bytes. So then I'm going to plot the original data here in these black circles, and then I'm going to plot the predicted data in green. What I found when I ran this model is that the p-value is 0.267. This is not significant. So snakes of any size can bite. Okay. Next, let's talk about linear regression. This is when you want to use number data to predict other number data. And for example, you might say, do larger fish live longer? These are the data that we're going to use. The left side is the fish size and then the age at death. So you can see here that these are both numerical data. These are both numbers. And so that means that they are continuous, right? We have a continuous factor, and then we have a continuous response. So our null hypothesis, remember, number data cannot predict other number data. So the null hypothesis says there's no relationship between these two values. How do we plot this? Well, we would use a linear regression. And this is the R code that I used to make the linear regression. First, I added fish size. Then I added the age at death. And then I made a plot of the age of death as a function of fish size. And that gave me these black circles here. Then I created a linear model using LN. I said, how is the age of death affected by fish size? And then when I asked the computer, what is the summary of this model? It gives me the formula of the model, and it tells me my p-value here, it's also here. The p-value gives me 0.00079. So that's very, very significant. And you can tell that by the number of stars that are here. And then it also gives you an adjusted R squared. So that tells you how much of the data you can explain using your linear model. So closer to 1 is better. Okay, so we've got an R squared that's very close to 1, that's a good fit. And then the p-value is very small, so that's very significant. Okay, so we've covered 4 or 5 different types of statistical tests. And you know now that the discrete responses and discrete factors, you should use Fisher's exact test. For continuous factors and discrete responses, you want this logistic regression test. For discrete factors and continuous responses, you want a t-test or an ANOVA, depending on how many factors you have. And for continuous factors and continuous responses, you want a linear regression. Okay, that's the end of today's lecture, but please send us more questions. If you have more questions about this lecture, please write them in the comments and we will do our best to answer them. All right, thank you very much. Akun charan.